Hello, this is going to be the videos for blood or hematology. That is the section we were going to cover this week, week 2. So I'm going to start with this first. I'm going to explain what's going on in this tube, and then these values. There's a few of these you need to know, and there is a PowerPoint that has these. I'll go through that with you. But first, let's just talk about what's going on inside this tube. If you want to add some anticoagulants, if you put blood into a test tube, you can do two things. You can either add some anticoagulants to where the blood will settle on the bottom and not clot, and the plasma ends up on top, or you can centrifuge it, and you can see that next. If you've never seen a centrifuge, and this is the picture that is on our first page and it's in our PowerPoint as well, you have blood that is separated. The plasma's on top, the little buffy coat's kind of towards the bottom, well past the middle, and then you have the blood at the bottom. How you do that? You can do it through chemicals, or you can use this type of machine. Now what she's putting in there, those are blue weights to keep everything balanced, and now she's adding what looks like 4 vials of blood. You hit the little switch, it's going to spin it around centrifugal force, which is why it's called a centrifuge. And then it's going to separate the components of blood, so then you can view the plasma, the buffy coat, and the red cells at the bottom. So you saw in that video, the little machine spins it around. What ends up happening is the plasma ends up on top. This little white layer kind of represents all of the white blood cells, and then the red is the red blood cells. Now these are called the formed elements, and I'll give you these percentages here in a second. But let's talk about plasma first. So first thing you want to be familiar with is what is the amount, what's a normal volume for plasma in a blood sample? And here it is. Plasma is about 55% of the whole blood volume. And then you see this 100% here, and then down here this represents what are called the formed elements, and it says here less than 1%, but pretty much the formed elements are 45%. So that's the first thing you want to note. How much of plasma is normal? It's about 55%. It's going to vary with a person, whether they're hydrated, genetics, but that's the golden number. So if you go up here into this area here, it says percentage by weight, and then you have this list over here. This brown represents all of these things that can also be floating in plasma. Albumins here, those are actually little proteins in blood. They're carriers. They help maintain the osmotic pressure of blood. Probably the important ones are these two. Globulins are a whole category of substances. We're worried about what are called immunoglobulins, which have to do with immunity and antibodies, that kind of thing. And then fibrinogen is a clotting agent. Fibrinogen is soluble in plasma. You can't see it. It's not solid, but this is a clotting agent. You have to have fibrinogen or else blood does not clot. So let's break this down. Now, what I'm about to show you, this blue area represents water. You need to be familiar, need to know how much of plasma is water, and we're going to use this value right here. 92% of plasma, you see it here, is water. The other 7%, I kind of just touched on these, and then you have all these other things that are sort of dissolved in, which would kind of represent like a 1%. But this is everything in plasma, so you have electrolytes, water, proteins, waste, nutrients, gases. This is nitrogen, oxygen, carbon dioxide. Those are called dissolved gases. So in this blue area here, if you have this out or however you want to study for your exam, you need to know water is 92%. These other substances, it says other solutes 2%, somewhere in there. I know that maybe the math, so you could say maybe this is 1, even this might be 1, but ions, nutrients, waste products, gases, I showed you that, oxygen, carbon dioxide, those are all things that are dissolved in plasma. So here's the big take from this. Plasma is 55% of a blood sample. Fibrinogen is a clotting agent. You got to have it for blood to clot. And then this water area, the blue, represents water and plasma, which is 92%. Now, if you go down to the bottom, the formed elements here, 45%. And then here's the breakdown. Now, these areas here, it kind of represents, and you have to remember platelets or thrombocytes are also part of this, but these represent all of the white blood cells that are found in the formed elements. We're going to go over those a little later. The hierarchy here is, once again, if you're watching this video and you want to write these down, neutrophils go here. They're the most abundant. Lymphocytes are the second most abundant. Here you would put monocytes, the bigger cells. Here are what are called eosinophils, and the very bottom here would be a basophil. That's the order that they go in. And as far as the numbers per cubic millimeter, I will cover that next. Okay, now in regards to, let me make sure I hope I'm not trying to make you dizzy. On this area here, you have to know the values for what is a normal blood cell count, a CBC. That area I've decided represents platelets. This area here is representing the white blood cells, and this represents how many red blood cells are in the formed elements. If you start with this part up here first, if you go to your outline, and I'm trying not to bounce back and forth, all right, a normal count for platelets here is about 250,000 to 500,000 per cubic millimeter per mm3. A normal white blood cell count is this one. It is 5,000 to 10,000 per cubic millimeter per mm3 of blood. So this is 250 to 500,000. This would be 5 to 10,000 per cubic millimeter. And as far as red blood cell goes, they are small and they're going to be the biggest number. You're talking about in the millions. And I'm trying not to bounce back and forth too much. But if you go up here into your outline in this area, 4.8 million to 6.5 million per cubic millimeter. That's going to vary, but depending on how big you are. And then a hemoglobin count is 14 to 18 grams per deciliter. So this little area here, that's a normal amount of hemoglobin within a given amount of blood. Okay? We will continue on next. Okay, so here was the white cells. This area here represents, I'm sure it's supposed to represent platelets, and this would be the red cells, which is called the hematocrit, and we covered all that. And this is a hemoglobin molecule, covered that as well. Okay, so before we leave this, 55% plasma, 45% called formed elements, which represent cells. And in a centrifuge, them being heavier, they're going to be pushed towards the bottom. Okay, just covered this. Water, 92%. Here are the things that you find mixed in with plasma. Albumins, globulins, fibrinogen, which is probably the most important thing here at the moment. It's a clotting agent. Without fibrinogen, blood does not clot. Okay, so under erythrocytes, let's pull up a picture of an erythrocyte. And I had shown this in lab, what this is before we get there. This is the entire blood cell formation. This up here is called a stem cell, and that cell has the potential to become any one of these blood cells. We're about to talk about erythrocytes. You don't have to know about this little flow chart. If you take histology, you will, but at the moment we're not worried about all the different levels of cells and everything, but one stem cell could become any one of these. So here is, I'm sorry, I'm going to make this bigger. So here's a red blood cell. This structure, the outside is thick, The inside is thin, so that allows them to bend. That shape actually puts hemoglobin closer to oxygen. And a couple of terms you need to be familiar with. Oxyhemoglobin is when oxygen is bound to hemoglobin. Deoxyhemoglobin is when there's less oxygen or no oxygen that's bound to hemoglobin. And on this little hemoglobin molecule at the bottom, hemoglobin has 4 little binding sites. It can carry 4 oxygen atoms. So, and like it says here, it's able to squeeze through capillaries, but oxyhemoglobin, if blood is loaded with oxygen, it has a red color. The deoxyhemoglobin, blood that's low in oxygen, it's not bound to hemoglobin, actually has a blue color to it. So I'm going to bounce back. Yeah, they are a biconcave disc. Like I said, this is hemoglobin. It's kind of showing the little binding sites there. And then oxyhemoglobin, deoxyhemoglobin, yes, it has a red color to it. Blood that's low in oxygen has a blue color to it. Normal red cell count. Kind of already covered this. That's just a sort of a range. It's going to depend on the person's body size. Children, it's going to vary. A lot of times males are bigger than females, so they may have more red blood cells. Guys, Shaquille O'Neal, 7-foot tall guy, is going to have more blood than I do. This would be the normal range per cubic deciliter for hemoglobin, 14 to 18 grams. Depends on your genetics. And then the lifespan of erythrocytes, they last about 120 days. They start bending down. They'll get old. And at that point, they get absorbed and broken down, and then everything gets reused. Now, I'm not going to draw this, but here's what happens when a red blood cell is broken down. The part that's called heme, because it's hemoglobin, and you can barely see it on our picture there. It says heme group. If you can look at that a little closer after this video. The heme part is broken down in blood. So like it says here, first of all, it's split into heme and globin, and the heme part is broken down into iron and a green pigment called biliverdin. These are called bile pigments. And the iron is reused to make new hemoglobin. The biliverdin, this happens in the liver, is then converted into something called bilirubin. And there's a lot of steps to that, and we're just being very vague with that. And the reason this has this color, bilirubin is an orange pigment, and it's excreted as what are called bile pigments. It's actually what gives urine and feces their color. And yes, if a person's liver is behind, they're going to end up with more bilirubin not being converted into bile pigments, and it gets stored in their skin, and that's where jaundice comes from. As far as control of production of red blood cells, this hormone controls how many red cells are in a person's system. It's called erythropoietin. Synthetic versions referred to as EPO. This little diagram kind of shows what happens with this. The kidney controls red blood cell production. There are cells in the kidney that produce and release this hormone. They're called JG cells, and we're going to kind of go over that later with the kidney. But here is a physiological scenario. If somebody has lost blood, or they're in low oxygen environments, they're in altitude, any number of reasons that they don't have enough blood cells, they're going to end up, no blood cells means you're going to have low blood oxygen. The kidneys are going to sense that. They sense big changes in blood pressure. They're going to release, the cells there are going to release erythropoietin. It's going to travel to red bone marrow in the body, and then cause the production of more red blood cells, which would, more red blood cells, the more oxygen a person can carry. And then you're going to have an increase in blood oxygen. And then, yeah, at some point, erythropoietin will be cut off. Remember the whole negative feedback part. So that hormone controls red blood cell production. Okay, I'm going to quickly go through this, and then I'm going to stop this video and pick up with the more detailed part. But leukocytes, went over this in the lab videos. White blood cells. You do have to know a few things, like what's a normal white blood cell count. I kind of did that earlier. And then what's a normal count, and then what's called a differential count. So here are the categories, and I'm going to skip ahead. I'm going to go back through this one more time, but at the bottom, these are all the white cells we cover. These three are called granulocytes. These two are referred to as agranulocytes. So to go back up, so starting here, granulocytes have granules in their cytoplasm, and you can see them. First candidate is called a neutrophil. They are the most abundant of all the white cells. They are phagocytes, which means they go around and eat other cells, but they are the most abundant, and they have 2 to 6 lobes on their nucleus. If you cut your skin and microbes get into your system the minute you cut your skin, these guys are the first ones to arrive to start fighting infection and trying to remove any foreign microbes or anything from your body. So yeah, they are the most abundant white blood cell. And like it says here, phagocytosis means they eat cells, they ingest solids and break them down. They're little Pac-Man cells. An eosinophil, they have 2 lobes, so it's called a bilobed nucleus. They look like shades. They do kind of stain a reddish color, and there's the granules. They're involved with allergic reactions or parasitic worm infections. They attack parasites. So a person that has a worm infection, they're going to have a very high eosinophil count. Basophils are the least abundant of all the white cells. They're less than 1%. They're involved with inflammation. They release histamine, which causes capillaries to leak fluid, cause swelling, and they also release heparin, which increases blood flow. Those two together help with what's called the inflammatory response. If you have inflammation in your body, it means you have damage or an infection, and the whole goal of that is to try to reduce the infection. So that's a basophil. The 2 agranulocytes do not contain granules in the cytoplasm, even though it might look like they do. They really don't. See the clearing here around it. There's the big nucleus. This is a monocyte. They are the largest by size of all the white blood cells. They are also phagocytes. They go around and eat cells they don't recognize. They eat more than neutrophils. They can move in and out of the bloodstream and go into connective tissues and things, and they clean all those out. So that's a monocyte. Biggest cell as far as size goes. In a lymphocyte, they are involved with what is called specific immunity. And we're going to cover that with the lymphatic system. But yes, they are what are called B cells and T cells down here. T cells make up 80% of a person's immunity, and it's very specific. They only attack certain things. They attack basically whatever the stimulus was that caused them to be produced. If you got chickenpox as a kid, then you ended up with T cells that were specific for chickenpox and also B cells, and that's the only thing they're going to attack. And they get stored in your lymphatic system. B cells produce antibodies, these little Y-shaped structures, and they are 20% of specific immunity. So that's all of the white cells. So there they are. Neutrophil, see all the lobes, an eosinophil, bilobed. A basophil also kind of, the nucleus is sort of bilobe, but you can't see it usually because of all the blue dots that are in the way. Here's a lymphocyte. See the little white layer of cytoplasm, the big purple nucleus, and then this is a monocyte. See how big it is compared to the red cells? These are platelets, and these are red blood cells. Normal white blood cell count is 5,000 to 10,000 per cubic millimeter. If you have less than this, you could have a virus. It's called leukopenia, actually. If you have more of these, then you have an infection, leukocytosis. A differential count is a count of each individual white blood cell. How many you have of each individual one. So a normal count is a count of all of these. A differential count would be a count of each individual white blood cell. Leukocytosis is a number above 10,000, and then leukopenia would be a count below 5,000. Okay, I'm going to start here with the next video. Thanks.